Alrighty. All right. So one of the things that happens during evolution is that something's changing, right? So the difference between me and a banana is, in its simplest terms, how would you describe that? How could you sort of say what's different at the most fundamental chemical level? Not, my phenotype or anything, but what is different really? Why am I different from a banana? Say again? Living and not living. Well, it is a different kind of life. My life as a banana I wouldn't be here. Or that would be a really weird day for you. What I'm getting at is that it's the DNA, right? Like if I had banana DNA, I'd be a banana. Now, I'm related to bananas, right? Because there are differences in my genome, in the sequence of letters in the DNA. So I don't make banana proteins. I mean, I do actually make some proteins that are the same as bananas, because bananas have some of the same things that they need to do that I need to do, which is if they need to make more cells, they've got to copy the DNA. What are they going to use to copy the DNA? They're going to use DNA polymerase. So I have DNA polymerase gene, they have a DNA polymerase gene, it's almost identical. So if you're changing your gene to evolve into different things, right, then that's achieved by making little changes in the DNA. Do you think that all parts of the genome evolve at the same rate? No. What kinds of things would be changing more slowly? Say again? Well, thinking about when the mutations actually happen, they're pretty random for the most part. But when they're selected for or against, right, then that's going to determine which things evolve more quickly, which proteins are changing faster. So what kinds of proteins would you not want to make big changes to? Or if you did, instead of becoming a banana, you'd just be dead. Say again? Potassium? Can you elaborate? Potassium is an ion, so what do you mean when you say that? What's that?
I said never mind.
Oh, but there is potassium in bananas, so I kind of see where you're going with that. That's true, and there's potassium in me. And I can kind of take that to a place, which is that we all have some similar conserved requirements, right? I need potassium to make my nerves work. Bananas need potassium for a different reason. But I have to copy my DNA. So I'm not going to go around messing with my DNA polymerase or, other machinery that everybody has too much, okay? But I could, for instance, toss out the lactase enzyme, and then I just have lactose intolerance, but I'm not dead, right? My point is that if you want to understand relationships between very distantly related organisms, what you're going to want to do is look at genes that don't change very much. Now, if you're thinking about categories of things that change rapidly and it doesn't matter too much what you do to them versus things that change very slowly, where do you think ribosomal RNA is going to fall in that spectrum? Slow, right? It's a machine that does this thing that all cells do. And they pretty much do the same thing that they've been doing since the beginning of time. So you can make small changes, and we know this because we know that ribosomes in eukaryotes are different from prokaryotes. So that you can make some changes and it's not going to destroy everything, but those changes will be slow, and there are going to be very few of them that you can make without having a big problem. So what he was doing was looking at ribosomal RNA, and as a result of that, he was looking at a bunch of different prokaryotes, and he saw that the sequence of ribosomal RNA between different prokaryotes differed more than he expected it to. And he was able to classify different things into different categories and said, oh, well, you know what? Based on the sequence of the ribosomal RNA, there's clearly a group of organisms, we're going to call them bacteria, and guess what? They also have some structural similarities, like they all have peptidoglycan, versus this other group that doesn't have peptidoglycan, still a prokaryote, and is, in fact, it turns out, much more similar to us than bacteria are. Okay, so here is a summary of some of the important features of these three kingdoms. So if we're looking at archaeans, then they are prokaryotes, meaning they have no nucleus. They don't have peptidoglycan. Instead, their cell wall has other things in it. And in comparison, bacteria, which are prokaryotes, do have peptidoglycan in their cell wall. And then eukaryotes are different again. If they have a cell wall, what's in it? It's not peptidoglycan. It's not like what the archaeans have. It's basically carbohydrates. It's basically glucose-based. It's either cellulose or chitin. Now, we've talked about this before, but this is just sort of to remind you of sort of the really big picture of how these three domains are related to each other. The idea is that the common ancestor of all three domains is about 3 billion years ago, when that early cell existed. And about 1.5 billion years ago, the bacteria and archaeans diverged from each other. The eukaryotes are thought to have been derived by an endosymbiotic process where a larger prokaryotic cell engulfed smaller ones and started this process of making organelles, basically. And there are some, as I've talked about before, some living examples of this kind of endosymbiosis that might mimic the way this would have happened early in evolutionary time. This you may be somewhat familiar with, or perhaps not, but these are the levels of classification. And so in other courses, you may have had to memorize them. You do have to, in this course, if you want the 1 point on the one question I might ask on the one test. So I might ask you if these classifications are put in the correct order. Since the whole chapter is about classification, it behooves you to actually know what the classes are and what order they appear in. So species, genus, family, order, class, phylum, kingdom. And I'm sure there's some fancy mnemonic that you could use to remember this. I don't have one to offer you. Usually I do, but in this case I don't. But I'm just going to warn you now that it's extremely likely I will ask you that type of question. All right, I'm going to focus today on species because the definition of species is different in eukaryotes versus in prokaryotes and in viruses. So, how do we classify eukaryotic species? Anybody know this? If you were talking about snakes or frogs or people, how can we tell if we're a different species? Can we tell just by looking? So this is the definition in eukaryotes. It is a group of closely related organisms that can interbreed. So basically it comes down to sex, right? If you can have viable, fertile progeny with another organism, then that is an individual that's in your same species. Here's an example of speciation within the group of bluebirds. These are all very similar looking but distinct species, and we know that they're distinct because you can't breed them and have viable offspring. If there were other hominids still living, then we would know that they were or were not part of Homo sapiens on that basis also. On the other hand, the prokaryotic world, we can't use that same definition. Why not? What's the the prokaryotic sort of equivalent of sex is this conjugation which may or may not ever happen. And in some bacteria don't do it at all. So, and most of them don't do it very often, even if they do. So it would be a really terrible way of defining species. I think the way that we define it in the textbook is kind of pretty terrible also, but here it is. A population of cells with similar characteristics. I don't like this definition very much, and I don't like it in part because in addition to being super vague, it's not really what's being used currently, okay? So actually, currently, the way we define species has to do with sequencing the DNA for the most part. But I'll get into those details in a second. Some terms that you need to know: culture, clone, and strain. So when we're working with stuff in this lab, we culture things, right? And that's just basically, it could be a mixed culture or it could be a pure culture. So what is an example of a mixed culture that we have cultured in this lab? When did we do a lab activity where we had a mixed culture or inoculum had several things in it? Yeah, when we did our environmental survey. And when we have used pure cultures, that's basically every other experiment we've ever done. A clone is a population of cells derived from a single cell. So what would an example of a clone be that you've seen? Yeah, colonies on a T-streak. Each colony would be a clone. And then this word strain is that some of the cells, even within a clone, can differ genetically from each other if during the process of cell division, the copying introduced a mistake, which does happen. And so it's actually, a strain is basically a variation within the same species. Now, a lot of times the way that we sort of functionally define species is that we're working with pure cultures, and we know for sure that they're related enough to each other to sort of be considered the same species, even if some of the cells within that culture are differed in some small ways. So when we classify prokaryotes, we use two different ways. We use phenotype, that is either their shape, the way that they do chemistry, or we can do it clinically using differences that we can detect using serological techniques or resistance to various kinds of drugs. The other way we do it, and this is much more common now in research, this is sort of done a lot in the clinic. This is done in research primarily, and it's genotyping. So we can genotype different prokaryotes in a variety of ways. I'm going to talk about 4 ways. So DNA fingerprinting, which used to be very common and now isn't as common. GC content, same thing, was common. DNA hybridization and sequencing, and not necessarily sequencing the entire genome, but sequencing, for instance, the ribosomal RNA. All right, so first let's talk about morphology. This should be somewhat familiar to you. What kinds of morphological categories do you know about? So how big the cells are? Shape. And what kind of shapes are common? Yeah, sorry about that. What kind of shapes do we? Rod. Cocci. Anything else? Spirals, right? Like that Lyme disease. So a variety of shapes and size and shape basically pretty much, and then also things like cell wall composition, right? Gram status, acid fast. Does it have a flagella? Does it have a capsule? Does it have, does it make spores? Those kinds of things. So we can do gram stains, for instance, to clump things into either gram positive or gram negative, which does narrow down the identity, and we're going to do that today. We can also do other stains to identify either chemical or structural features. So what kind of stains could we do? Endospore stain to look for endospores. Acid fast to look for mycolic acid. Capsule stain to look for capsules. And the other thing we can do is look at their metabolic abilities. Okay, so what kind of metabolic tests could we do that we've learned how to do? We could do a fermentation test. Yeah, what kind of enzymes have we tested for? Yeah, caseinase is one way to call that enzyme. Anything else? What are the 2 instant tests we could have done? Oxidase, catalase. Right? We also have that MRVP test, which is looking at a subtype of fermentation. We can also look at salt tolerance, right? So there's many different things we can do, not just the ones we've done in lab, but those are relevant. We can also look at their growth requirements, so what they might need in their medium, which we really haven't done. Look at whether they can live on complex or defined media, and we can also check things like tolerance for salt, or, as we'll do today, we can look at their requirement for oxygen using the DEEPs. So what is this here showing you? It is a dichotomous chart which you created today or sometime earlier this week. And it can be used as, we're going to use it to, as part of this exercise. In the clinic, it could be used as a quick diagnostic. If you have a patient who has sort of telltale signs of something like diarrhea, and we know that there's like only a few very common possible causes. I mean, it could be something you've never seen before, but far more likely it's something you have. Let's say there are 6 things, right? So you can narrow it down by just doing a couple of really quick metabolic tests, and you can be fairly sure that you know what it is, right? So if you have a patient presenting with diarrhea, you could first do an oxidase test, because that would for sure, that would sort of confirm the possibilities that you're thinking about. You could then do a lactose fermentation test, which eliminates these 2 Enterobacter and Enterobacteria from these pathogens. And then you could do a citrate test, and based on the citrate test, that would narrow this down further. Now, it's not completely 100% certain, right? You could be wrong. But if you're dealing with a situation where it's a high degree of likelihood that there's, usually people who get sick have one of these 4 things, then we can narrow it down using this kind of testing. Commercially, these kinds of tests are done to look either for pathogens or for industrially important organisms or contaminants. And again, it's a context thing, right? You may be in the dairy industry and you're either looking for the presence of something that usually should be there, or you're looking to make sure that there isn't something there that is a common contaminant. And what people will have created this series of tests that have 15 or even 30 different metabolic tests in a line, and all you have to do is take a swab of the whatever it is that you're testing, and you shove it down this hole that goes into all the tests, and then you can get your answer like really fast. So that would have made your lab a lot easier, but we don't do it that way. So here's an example of what that looks like. So there's I think 14 different tests or 15 different tests. And you just inoculate them all simultaneously. You do the incubation, you read the results, and then based on the number that you get, you have a high degree of likelihood that it is some particular organism. So could you identify an organism based on this type of testing? Sure, because there's like 1 million species out there and we know 6,000 of them. But probably you're going to be right if you're in a context where it's very common to see certain things. Now, one way of identifying sub-strains of the species is with serotyping. And this is something that was pioneered by Rebecca Lansfield in the 30s. And it was really groundbreaking because at that time, people were getting sick with a lot of different sub-strains of Streptococcus pyogenes. And she did this serotyping technique that helped to specifically narrow down which things people were getting sick with. So how it works is that it relies on antibody-antigen interactions. One of those words might be more familiar to than the other. So what are antibodies? What is an antibody? In what context do we have them? Anybody? Do you have any antibodies?
Yeah.
Against what?
All types of diseases.
Okay. Where did it come from? Where is it in your body? Say again? It is a protein. And where is it located, usually? There's some in the mucous membranes, in the bloodstream, in the lymph tissue. Basically, circulating antibodies, they're not the only kind, as we'll see later on this course, look kind of like that. They're in your bloodstream, and they're looking for some specific virus or some specific bacteria that they've seen before, and they help you to remember things you've seen and help to fight them off. Now, we'll go into this in a lot more detail, but antigens are the things that the antibodies are looking for. The antigen isn't the whole organism. If this was a say, bacteria like E. coli, there's stuff pointing out of the E. coli that is detectable by antibodies. So that would be the antigen, and it has a specific binding to an antibody. So in our blood serum, we have circulating antibodies. That's not the only place, as we'll talk about in a couple of chapters, but it is where a lot of them are found. And in particular, if you're trying to determine if someone has already been exposed to a particular illness, you may be looking for antibodies against the thing that they were exposed to, and that's going to be in the blood, so you'll do a blood test. And so how are antibodies made? They are, this is actually sort of a misstatement. It's a very simplistic view that we will then deconstruct in a couple of chapters, but basically antibodies form in reaction to something foreign coming into the body. And that foreign thing may be something that is on the cell wall, on the capsule, on the flagella, or on the pili, something that's sticking out of the virus or the bacteria or whatever it is, the pollen grain. Now, if you just think about bacteria that have cell walls and maybe capsules, flagella, and pili, are these all identical in all different bacteria? No, there can be subtle differences. So let's look at an example. Well, we won't look at an example. First, we're going to look at the test itself. So this is what the slide agglutination test actually looks like. This is a positive on the left and a negative on the right. And what you do is you just take the antibody and you mix it with the thing that you're looking at, and you just see if the antibody has a reaction to that thing. And so on the left you can see that there is clumping, so the antibodies are binding something and basically taking them out of solution. So let's look at what some of those antigens could be. Here's an E. coli, and it has a plasma membrane, it's got a cell wall, and does anyone remember what gram status? Negative. It's gram-negative, so it has a gram-negative cell wall. It's got these flagella, and also it has a capsule. Now the O157H7 strain of E. coli has the 157th type of O antigen in its cell wall, in the lipopolysaccharide layer, that outer lipid bilayer. And it has the H antigen in the flagella. It's the seventh type of H antigen in the flagella. So the flagella all have a basically similar structure. They're all made of flagellin, but there are subtle differences that are detectable by your immune system. And we can also use serology testing to find those differences. When we have a difference between, say, O157H7 E. coli and some harmless E. coli that's just living in your gut very happily, then that's considered different serotypes because you can actually use antibodies to detect them. You can identify species and sometimes subspecies differences using this technique. There's another way to broadly classify and find strain differences, and that is that different species and even strains within species have different antibiotic resistance profiles, right? And we know this is true because, for instance, we know that penicillin works against what general class, generally speaking? Gram-positives, okay? And generally speaking, gram-negatives are resistant to penicillin. But we also know something like Staphylococcus aureus, which is gram-positive and in its normal state would be penicillin sensitive. There are some sub-strains, right? Some strains of Staphylococcus aureus that are resistant to some versions of penicillin. So, for instance, what Staphylococcus aureus that you know about is a really big problem in a hospital? MRSA, methicillin resistant. Methicillin is a derivative of penicillin. So even though it should be penicillin sensitive, it's not. And that's based on genetic differences. So we can identify those differences by basically putting a lawn of cells down. So we put so many cells down that they're just touching each other. And then we put different antibiotic soaked discs on the lawn and you grow it up and see if the cells in the neighborhood are killed or they're not, right? And clearly, these are either different species or they're different strains of the same species. And again, context is everything. If you're in a hospital and you know what 16 things are likely to be floating around out there, this can help you to figure out which one it is, even though you might be wrong, but you're probably not wrong. A similar kind of technique can be used that has to do with the fact that bacteriophage, which are the virus that infects bacteria, also have specificity to different species and also even to different strains within a species. And so you can take a whole bunch of different bacteriophages and you can put a lawn down of whatever you're testing, and then you just squirt the phage into different parts of this grid and see which ones are able to get in and kill that particular species or strain. Now this was a very common method in the past. It's largely been replaced by the molecular techniques that I'll talk about now. And that is this list, including DNA fingerprinting, hybridization, GC content, and sequencing. So this is DNA fingerprinting. I am going to briefly introduce to you, it again was a very common technique that's been largely replaced by sequencing. So in order to understand this picture, we first have to draw another picture. All right, so remember that here's our prokaryotic cell, here's our DNA, and I introduced you to this concept of a plasmid, right? Does anyone remember what a plasmid is as compared to the chromosomal DNA?
It's like a chunk of the DNA outside of the chromosome.
Yeah, it's a small circular DNA. It's similar in concept to chromosome, but way smaller. And so whereas this could have thousands, or even in some cases tens of thousands of genes, this may have 3 or 4 genes on it, so it's really small. So I'm going to use that as an example because that is how that picture was taken, the one that you saw in that gel just earlier. And then you can sort of extrapolate that to a larger chromosome. So if we think about a plasmid, and let's suppose we just think about one that's kind of small, right? Then what this plasmid is it's a double helix, and each strand has, some sequence on it, right? And on the other side, there's also more. So you can just imagine that sequence runs all the way around. Now there are these enzymes called restriction enzymes. Has anyone heard of these? Okay. If you ever work in a lab, as I have done for 2 decades, then these will be like your best friends and your worst enemies. So restriction enzyme is actually made by different bacteria to kill their enemies. And what they do is the restriction enzyme chops DNA, but it chops DNA at specific sequences. So for instance, there might be restriction enzyme A, and it looks for this sequence right here. And when it sees that sequence, it chops the DNA. And so if this organism, which contained this restriction enzyme A, wanted to kill its friends or wanted to kill its enemies but stay alive, then what it would do is just evolve so that it didn't have this sequence in the DNA. And what happens then is that it can make this restriction enzyme, and everybody around who is not prepared for that will get killed, and it will live. So we've exploited that to basically use it as a primitive sequencing tool. So we just categorize all the restriction enzymes from all the organisms that we can find, and then we know if we wanted to cut this sequence, we can use restriction enzyme A. If we want to cut this sequence, we use restriction enzyme B that's specific to these letter sequences, okay? So that's the concept of DNA fingerprinting. So I'm going to show you an example of what that would look like, and then we'll go back to that picture so it'll make more sense. So here's my circular plasmid, and let's suppose that restriction enzyme A cuts here, and restriction enzyme B cuts here, and restriction enzyme A cuts there again. And we can also designate these pieces that would be cut by counting the number of letters between the different restriction sites. And let's suppose this is, there's 1,000 letters between here and here, and there's like 500 here, and maybe 400 here. So if we were going to cut this plasmid with both restriction enzyme A and B, then what we would get is a piece that was 1,000 base pairs long. We would get a piece that was 500, and we would get a piece that's 400. Is that clear so far? So if we chop this at these particular sites where these particular enzymes like to chop, then we get these pieces, and then it's in a solution, and it's invisible, and it's really small. So what do? We run it out on a gel, Have you done gel electrophoresis in your previous biology class of any kind? All it is basically, so you want to imagine these pieces of DNA, now they look like this. So this one's 1,000 letters long, this one's 500 letters long, and this one's just a little bit smaller. And again, it's all in a solution. It's in water, and you can't see it. So what we do is we make a gelatin that's flat, and it has these little wells, and we put the DNA solution in there. And then because DNA is negatively charged, why is DNA negatively charged? What are the nucleotides made of? What are the 3 parts of nucleotides? We've got our base and our sugar, and what else? Phosphate, which is a super negatively charged. So the whole thing's negatively charged. So we just put this in here, and then we run an electric current where the positive electrode is here and the negative is here. So these negatively charged DNAs are going to want to run down to the other side. But this is a gelatin, and the pores of the gelatin, we pour it at a certain percentage so that it makes it hard for the DNA to run to the other side. And the bigger it is, the harder of a time it has to get to the other side. So things that are big, like this one, are going to run more slowly. If we did this for a certain period of time, they would only get this far. And these smaller ones are going to run faster. Okay, so we basically move them through this current so we can separate them out by size. They're still invisible. And then we add a fluorescent dye that binds to DNA, and we use a UV light to see the bands. So that is how you end up with this picture. What's been done here is that different DNA plasmids have been chopped by different restriction enzymes, run through by electrophoresis, run through a gel, and then we added this dye to make them visible. And what you can see is that there's different sizes of DNA pieces. Okay, so I'm gonna try to help you to understand what you're looking at. So plasmid 1, which is shown in these 2 lanes in red, was cut in 3 places, giving you a band that is big, medium, and small, and you can see they're the same. And another plasmid was run and cut, and it was also cut in 3 places, but you can see the bands are a little bit different in size. So that kind of technology is how you fingerprint the DNA, right? This plasmid has this signature band size, and this other one has a slightly different size. And when you run it out, you see this pattern, and you can recognize the pattern. Now, this is just an example of a plasmid that's small. You can do this with a whole chromosome, and you get a ton more bands, lots more than this. But basically, you look at the bands, and you compare this to that, and you can say, oh, here's a lot of similarities, but here are a few differences. Now, there's clearly some disadvantages to doing this. It's actually really useful, much more so than you would imagine based on this abstract description. But it doesn't tell you exactly what the sequence of the DNA is, and so you could miss, there could be reasons that you could get these same band sizes and have different sequences. So it's not perfect. GC content is another way to just compare different genetic sequences. And so this is based on this idea that when you have base pairs, remember what pairs with what in the DNA, in the double-stranded DNA? G and C pairs with each other, and A and T pairs with each other. And what do you remember about the pairing? How does it work? Hydrogen bonds. And the number is different, right? G's and C's go together because they can make 3 hydrogen bonds. And A's and T's go together because they can make 2. And which one of those bonds is then stronger? G's and C's are being held together. And so if you heat the DNA because you need to melt the 2 strands apart from each other, then what's going to be harder to melt? G and C. So if you had a sequence of DNA that was all G's and C's, it would take more heat to take those 2 strands apart than if it was all A's and T's. And so the percentage of GC content will determine how much heat you need to completely separate the 2 strands from each other. And what this is a melting curve, and it's just plotting the point at which the strands have come apart and at what temperature that happens at. And so if you have 2 organisms that are super duper closely related, then their sequence is going to be very similar. So their percent GC content is going to be similar. And if you have two species that are very different from each other, then their GC content may be different. All right, so this technique is important to know, can be used to say that organisms are different, right? If you're taking the DNA out of one bacteria and out of another one, and the GC and the temperature of melting is really low in this one and really high in that one, then you can be confident that they're not the same species, right? Because that suggests that the percent GC is very different. Can you use this technique to say that organisms are very similar? The answer is no, because I'll give you an example. Let's suppose that, here's one example, and I'm going to make the genome really small. Obviously, it's got, hundreds of thousands of letters in it, but we're going to just do a few. So this is organism A. Okay, and this is organism B. So if we melt them, which one's going to melt quicker? Right. So this one's going to melt, and it's going to be like, let's suppose it's a high temperature and this one's a low temperature. Then we know clearly they're different. We don't know what this sequence is, though, right? So here is organism C. It has the same percent GC content as organism B. It's going to melt at the exact same temperature. But the sequence is not the same. So we don't know, based on GC content, you would then say they're identical, and we can't know that because we could just shuffle the letters and we would get the same result, but they're not the same species at all. Does that make sense? Okay, DNA hybridization is a sort of similar idea. It's actually more specific, though, because what you do is you take one organism and you heat it till the strands completely come apart. You take another organism and you heat it till the strands completely come apart. Then you mix them together, and if organism A and B were identical or really, really similar, then when you did that and you allowed them to come back together, they would almost always form a perfect duplex, right? Because all of those sequences would be similar, they would just sit together. And so it would look like this. And that would tell you, oh, those two guys are super similar or the same. If they're not similar at all, then you're never going to get any hybridization. So you can say for sure they're not the same. And if they're sort of similar and sort of different, then you're going to get some hybridization. And you can actually calculate how much as a percentage, and that will tell you how similar they are. So this can be actually used to tell things apart as different or similar and give you a number that tells you how similar they are. So this is really helpful. And until sequencing was common and inexpensive, this was a very powerful technique. But now we have this explosion of technology that has made it possible to get the letter-by-letter sequence of anything we want for not very much money. And so people are just going to town. They're sequencing everything they can get their hands on. And as a result, there's lots of microbes whose whole genome is available, lots of other animals and plants and fungi as well. And it's allowed us to take a much more detailed look at the whole of life. The downside to sequencing is that you still have to culture the organism and get lots of cells in order to do this. And so it is possible to do an ID based on a partial sequence. And to do that, we have to use a technique called PCR. Have you heard of this? Has anyone heard of PCR in any context? Have you ever watched CSI or anything else like that? Whenever they turn the lights off, which I don't know why they do, it's crazy, and they have a pipette and that looks sexy, which is not true at all. It's not sexy to hold a pipette. And they put their glasses on and they're playing with tubes. Most of the time they're doing PCR. It's an incredibly widely used clinical technique. And it's basically asking sequencing basically would be like this. So here's our prokaryote, here's our chromosome. Now let's simplify our chromosome so that we can actually understand it a little better. Let's pretend this is our chromosome. Sequencing would be if you read every single letter, and then you knew exactly what was there, okay, for the whole chromosome. That's sequencing. But what you can do instead is you can ask a question about this region right here that you happen to know is there, and just what's the sequence of that, or is it there at all? So you can ask a smaller question without having to read the whole sequence, which is handy because if you want to do this, if you want to know what the whole sequence is, you have to have a lot of DNA. And that means you have to culture the organism. But if you want to ask this small question, we can actually use this technique of PCR to ask that question, even if you have only the tiniest amount of DNA. And how that works is basically, have you ever watched a show when they were looking at like crime scene and they get a little bit of DNA from a blood sample or from a hair sample? There's only the tiniest amount of DNA. It's not like you can take that immediately and sequence it. The process of sequencing it would destroy it. So instead what you do is you use that in a PCR reaction to amplify the amount of DNA so there's tons of it, and then you can use that in sequencing. Okay, so how does that work? I'm going to show you on this picture and then we'll walk through it because it's easy but complicated. All right, so the idea is that if this is, say, the whole DNA of some unknown organism, and we don't have the ability to culture it, so we can't get lots and lots of this, but we know that probably in there is a little sequence that looks like this. We can artificially make this sequence, and it's going to bind to this DNA, and then we can use PCR to make lots and lots of copies. So how does that work? So again, I'm going to use this example as if the DNA of an organism is really small. Okay, so let's suppose that's the whole genome, which would be ridiculous. Okay, so that means that this is what the genome really looks like. There it is. And let's suppose that we want to ask whether this part here actually is in there. I'm going to have to make this a little bigger for it to be. Okay, so we want to see if that's there. So what we can do is we can make primers to this region that we know should be there, and we can amplify up the DNA. So what does that mean? Remember that by convention, this is the 5' end and the 3' end. So what does that mean? What is the 5' end? What's sticking off up here? Your phosphate group, right? And over here is that 3' hydroxyl group. So if we melt the DNA, and we have primers. What are primers? Do you remember when I was talking about DNA replication and I told you that the DNA polymerase can't just start replicating without being kickstarted by an RNA primer? We can do something similar. We don't use RNA, we use DNA. But we just make a little DNA primer that's going to sit down here. So we make this sequence right here. And I'm going to make it in pink so you can see that it's not the original sequence. So there's our primer, 5' to 3'. And this is an end the DNA polymerase can use to attach to. We have another primer over here. Again, it has that hydroxyl group, so this is the 5' end over here. And so we just make these little pieces of DNA. We get this DNA from our unknown. We don't have very much of it. We melt it. We add these primers. We add a whole bunch of nucleotides. And we add DNA polymerase. And we add some ATP. And then we just let the DNA polymerase do its job. So it's going to replicate this DNA like it always would, right? So we put this all in a test tube, and then we just let it go to town. And it's going to make this copy over here, and it's going to make this copy over here. And then, okay, so now there's 2 copies where there was just 1. Then we do this again. And again. And again. And if you remember how exponential growth works, if you do this 40 times, we have $1 million, right? So we go from 1 copy to 2 copies, to 4 copies, to 8 copies, to 16, to 32, to 64. And before you know it, you have 1 million copies. And that is enough DNA to sequence, okay? Now you have to remember that this is going to be in the context of a much larger piece of DNA. And so we can't really realistically PCR up the whole DNA, and we don't care. We're going to ask a question about something specific that's going to help us to ID this organism. If we had a question about this unculturable organism that was about evolutionarily relatedness, what might be a good thing to sequence then? How did Carl Woese assign relatedness when he was building those 3 domains? What was he sequencing? RNA, ribosomal RNA, which doesn't change very much. If you want to ask a different kind of question, maybe you know that you have one of possibly 3 very closely related species, then you're going to PCR up something that does change a lot, like maybe their lactose enzyme. Maybe this strain has a lactose enzyme or lactase enzyme, and this one doesn't. We're going to try to see if it has it. So we can use PCR in a number of ways. It's a very powerful technique to get a lot of copies where there was only a little bit of DNA to start with. And then here's an example of how it's done, for instance, in genetic testing. So let's suppose there's a patient with cystic fibrosis and a sibling that is unaffected. And we know that the cystic fibrosis region has some genetic difference that you can actually, there is a sequence. So instead of sequencing the entire genome from both patients, which is a lot of work, we can just use this PCR technique with primers here and here to amplify this region, which is not going to be in the unaffected patient. We run a gel and then we see that one patient has it and we were right about our diagnosis. Okay, so we just have to substitute that for microbe in, say, in the world where we're looking for the ability to make an enzyme or whatever. And again, this is in a context where normally if you could culture this organism, you would just do a lactose fermentation test or something like that, right? But this is a context where we don't have the ability to do that. We just have the tiniest bit of DNA and we want to find out something about it. Okay, so we already talked about this a little bit. This is in more detail what specifically Carl Woese was sequencing when he was comparing different organisms and reassigning their relatedness was the 16S ribosomal DNA sequence. So this is the gene that makes this ribosomal RNA that's part of the small ribosomal subunit. So I'm going to just go back here to show you where that is. So remember I told you that in prokaryotes there's this 70S ribosome as opposed to the 80S ribosome in eukaryotes? And then it has 2 pieces, right? The little piece and the big piece. The little piece is 30S and the big piece is 50S. And this little piece has several sub-pieces as well. It's got one piece of ribosomal RNA that on its own is 16S and several 21 polypeptides. So it's just that one ribosomal RNA sequence that he was looking at. Okay, so how do we classify prokaryotes? We can do it based on phenotypes, so we can look at their size, their shape, how they stain on various kinds of differential or special test stains. We can look at their metabolism. We can also use these new molecular techniques that make ID much more accurate. So, and we can also use that information to construct these relatedness trees. And this has really allowed us to understand something that was not clear before, which is, remember I talked about the difference between vertical gene transfer and horizontal gene transfer. Doing this kind of analysis has revolutionized our idea of how frequent this process is in prokaryotes, because we know it doesn't happen very often in the eukaryotic world. This is how you build one of these trees. So basically you just get the sequence for the ribosomal RNA, and you know, there's obviously it's longer than that, but let's suppose that that's really how big it is. And you look at the sequence of letters for each organism. And what you can see is that some of these sequences look more similar to each other than others, right? And you can actually run this through a mathematical algorithm that's going to tell you how similar they are and give you a number. So these 2 species, Lactobacillus brevis and Lactobacillus acidophilus, are 70% similar. So they're going to be the most closely related in time. And you just can make this little diagram where these guys are 70% related, okay? These guys, this guy here is 60% related to both of them. If you look at this numbers, Lactobacillus plantarum is 60% related to both acidophilus and brevis. So then you can put it over here. And then this guy over here is 50% related to all of those. And that's how you can sort of construct this relatedness tree. This is what it really looks like if you do it on a bunch of organisms. So here's just a sort of small subsection of the different kinds of bacteria that you can do this kind of analysis with, and you start to see, you know, these guys are closely related to each other and very distantly related to those guys. But it gets really complicated. So this looks sort of nice and neat. I mean, there's a lot going on here, but it does look pretty organized. But this is what you see when you do a ribosomal RNA comparison. A ribosomal RNA is not the only thing changing, and it is the part that's changing the slowest. If you look at a lot of other things like, say, antibiotic resistance, then what you notice is that antibiotic resistance gene is popping up all over the place, right? Because when we talked about horizontal gene transfer, it can cross species in even distantly related species. So if you do a different kind of analysis, then it looks like all kinds of crazy things are being passed around because of horizontal gene transfer. The most important thing to remember is how to read these kinds of evolutionary trees, right? So if I ask you questions like, species C, what's its closest living relative? What's that going to be? The living relatives are the ones at the ends of the arrows. These join points are like hypothetical ancestors, right? Where they diverge from some hypothetical ancestor, but this is what's alive now. So what's the closest living relative for C? D, okay? And the closest ancestor for both A and B, so here's A and here's B, what's the closest ancestor? That would be 5 over here, right? So just being able to read the nodes and then the endpoints of all of these is what you need to know. Okay, so that's all of chapter 10, basically. That makes me happy. We're going to move on to the lab now.